Hello and welcome back to Microbial Concepts. So in this video, we are going to discuss about isolation of photosynthetic bacteria. Now, I have included both theory part and the practical part. For few, this practical is in third year BSc, and for few you might have in masters. Okay, so according to that, whatever information is necessary for practicals or theory, everything is included here. So photosynthetic bacteria, when we uh, read about photosynthetic bacteria, first name that comes to our mind is cyanobacteria, right? But that is not just one example. There are many different photosynthetic bacteria present in our environment and we need to study them. So for that purpose, you have this practical in your syllabus. Now it depends that depending on the material available or depending on the syllabus, you might have just cyanobacteria to study, or you might have purple sulfur bacteria or green sulfur bacteria or purple non-sulfur bacteria, etc. So let's see. So photosynthetic bacteria are some of the most widely distributed microorganisms in the environment, playing an important role in our food chain, okay, amongst animals and plants, as plants are considered to be the base of the food chain, okay. Likewise, photosynthetic bacteria are also considered as the base of a food chain, okay. Now, these microorganisms that are capable of synthesizing some organic compounds using light as their energy source, they are very, very tiny, but they are very important. They are mighty. So they employ various pigments such as chlorophylls, bacteriochlorophylls, and carotenoids to capture light energy for photosynthesis. Now, at present, they are represented in three families with around 20 genera, and all of them are gram negative except Heliobacteria, okay, not Helicobacter, okay. This is Heliobacteria. This is the exception for photosynthetic bacteria. This is the one which is gram positive, rest all are gram negative. Now, the cell suspension of various species, they may occur purple, red, orange, brown, depending on the genus of the bacteria, depending on what the chlorophyll, what type of chlorophyll the microbe is utilizing. Okay. Now, the genera are subdivided into two main groups. Those are first as purple and another is the green. Now, they are all aquatic. They are abundant in the ooze of ponds, so they are easy to isolate and study. Now, these photosynthetic bacteria, they differ from algae and green plants in that the bacteria, they are anaerobic. Okay. Now, cyanobacteria is one which uses oxygen, okay, or aerobic. Rest all are anaerobic and they utilize bacteriochlorophyll instead of chlorophyll A for photosynthesis. That's the main difference. Now, why to study these photosynthetic bacteria? Yeah, they're tiny but mighty. That is the very important line here, because they are unicellular, okay, and they are the primary producers of our ecosystem, meaning the base of our food chain. They also help in nitrogen fixation, making it available to the other microorganisms or other organisms. Additionally, these these bacteria, they are studied for their potential use in bioremediation and the production of biofuels as well. Okay, that's the reason why they are important and they are included in our syllabus. Now, just to make your concept clear, what is the difference between chlorophyll that is present in photosynthetic bacteria and the chlorophyll which is present in plants and algae? Now, even when we term or we use the term algae, they are eukaryotes, and in bacteria we have cyanobacteria, which we also name as blue green algae. That is the only one which is prokaryotic bacteria. Okay, there are various examples in cyanobacteria class, but they are prokaryotic. Okay, so the type of chlorophyll that the photosynthetic bacteria they use, it can be A, B, C, D, E, or G. Okay, in case of plants, A and B are most common or primary pigments, and even we find C, D, and F. Okay, so primary pigment is A or B in bacteria as well as A in plants. Okay, now the absorption spectrum of photosynthetic bacteria, chlorophyll, is they absorb light in the infrared and some in the visible length. The chlorophylls in plants, they absorb light in blue and red regions. Okay. Now the oxygen production is the very important point here. Now the chlorophylls that photosynthetic bacteria they use, they does not produce oxygen. That means they are an oxygenic photosynthesis. Okay, they are used or they use an oxygenic photosynthesis. In case of plants they do perform oxygenic photosynthesis. That means they produce oxygen. Okay. Now the reaction centers are different in both. Then for examples, uh, if you get a question like what is the difference between chlorophyll in photosynthetic bacteria and in plants, give examples. Then in case of photosynthetic bacteria, you can give examples of purple bacteria, green sulfur bacteria, heliobacteria, etc. If you can remember the full name of example of a purple bacteria, okay, the genus and the species, that is also okay. Then in case of plants, you can just mention that the higher plants, green algae, cyanobacteria, they are used or they use the chlorophyll A, B, C, D and F. Okay. Now here also they have mentioned cyanobacteria here, but it is actually a photosynthetic bacteria. Okay. Now membrane location. Now the chlorophylls they are embedded in the intracytoplasmic membrane or chlorosomes, and in case of plants they are embedded in thylakoids or the thylakoid membranes of the chloroplast. Okay. Now about the types of photosynthetic bacteria. Now we know that they are a diverse group of bacteria that can harness energy of sunlight and produce their own food. Okay, similar to plants. Now depending on the reaction, photosynthetic reaction, they are categorized into two types, that is oxygenic photosynthetic bacteria. Okay, so these bacteria like cyanobacteria they perform photosynthesis in a similar way to plants, and they use water as electron donor. Okay, that's the main difference here, and they release oxygen as a byproduct. So cyanobacteria is an example of oxygenic photosynthetic bacteria. Another type is anoxygenic photosynthetic bacteria, where these bacteria they use different electron donors instead of water, such as hydrogen sulfide or sulfur, and they do not produce any oxygen. That's the reason why they are grouped separately. Now there are types in anoxygenic photosynthetic bacteria. For example, purple sulfur bacteria. Now these bacteria they use hydrogen sulfides as electron donors, and they are often found in freshwater environments. Okay, they get their name because of the pigment that they contain, that is bacteriochlorophyll. Purple non-sulfur bacteria are one which use a variety of electron donor including organic compounds and are often found in soil and marine environments, easy to isolate. Then green sulfur bacteria are one which use hydrogen sulfide as an electron donor and are found in freshwater environments. They contain green pigment called chlorophyll F. Okay, so they are termed as green sulfur bacteria. When they don't use sulfur, they use other organic compounds as electron donor, you term them as green non-sulfur bacteria. Okay, that's the only difference. Now, what's the difference between oxygenic photosynthesis and anoxygenic photosynthesis? This one you can get in your viva questions. So, in case of oxygenic photosynthesis, oxygen production is done. In case of anoxygenic photosynthesis, no oxygen production is done. What are the electron donors? Oxygenic photosynthesis uses water, and oxygenic photosynthesis uses hydrogen sulfide, sulfur, or other organic compounds. And examples are given here. Now, if you want few examples to remember for your Viva exam, so you can at least try to remember two or three examples. So first is very important, that is cyanobacteria. So from cyanobacteria, you can keep in mind oscillatory or Lingbaia. Okay. Then in case of purple non-sulfur bacteria, you can remember Rhodosudomonas palustris. Then Chromatium venosum is another purple sulfur bacteria. Okay. Then Chloroflexus aurantiacus is green non-sulfur bacteria, and Chlorobium tepidum is green sulfur bacteria. Okay, so each type I have covered here. So try to remember at least 2 to 3 examples for your viva. Now moving towards our isolation part, as this one is a practical related video. So our aim here is to isolate and identify a photosynthetic bacteria from given environmental sample. That can be soil or water. Okay. Now what materials you need? So sample say from a pond, you can collect the mud, you can collect water as well. You need petri dishes, petri plates, then Erlenmeyer flask for enrichment. You need inoculation loop. You need illuminated growth chamber, as we are isolating photosynthetic bacteria. So light source is required when you are incubating plates, and cotton plugs. Okay, so this is basic material that I have listed here. Now the type of media that you use. Now it depends on the desired microbe you are trying to isolate. For example, Bennett's broth here, it is actually used for purple sulfur bacteria or green sulfur bacteria, as it has sulfur in its composition. Others are light broth, Fennings broth or medium, and BG11. Now BG11 is commonly used for cyanobacteria. BG stands for B, blue, and G for green, blue green algae. Okay, that is cyanobacteria. So BG11 with nitrogen, without nitrogen source, uh, different compositions are present. So you can try those for specifically for cyanobacterial isolation. Okay. So moving towards the isolation part. So let's see, if you are using Bennett's broth for isolation of an oxygenic photosynthetic bacteria, that is say purple sulfur bacteria. So for that you need to prepare Bennett's medium, then you transfer it to 250 ml in 500 ml of Erlenmeyer flask. You plug and autoclave it. Then when the broth is cooled down, add 1 gram of the pond mud. Okay, in the flask, incubate it for say 15 days at 35 degrees Celsius, because it is an enrichment medium. It will enrich the given sample, and it will help the bacteria to flourish and grow. Now this incubation is to be done in an illuminated growth chamber. The lamp should remain on for 24 hours per day. Okay. Now examine the flask each day to look for color change. Okay. Now depending on the nutrient availability, you might see that the color change in the bacterial growth. Okay, so you need to observe that as well. Then after incubation, algal colonies, when they start appearing in the flask, in the broth, you make a hanging drop slide of organisms just to check what kind of bacteria are growing. Okay, examine it with a phase optics or of a bright field oil immersion, and try to see whether those are photosynthetic bacteria or other prokaryotic bacteria that are growing. Okay, then prepare Bennett's agar medium by adding 2% agar in the broth, and you autoclave them again. Now you put pure culture. Okay, to get pure culture, you put few cells of algal colonies with the help of inoculating loop in the surface, on the surface of Bennett's agar medium in the plate. Okay, and then incubate the plate at 37 degrees Celsius for a week in illuminating chamber. Okay, again you check for the isolated colonies under the oil immersion lens, okay, in microscope. Now this one is for just purple sulfur bacteria, because the Bennett's broth, it has sulfide present. Now if you are using any other medium, then the protocol may differ a little bit. Now a general protocol for cyanobacteria. Okay, let's see that. You collect the sample from say your nearby garden or pond, and you try to isolate cyanobacteria from that. Now you transfer a small amount of sample onto the surface of a sterile plate or petri dish containing the PG11 agar. Okay. Now place it place the inoculated petri dish in incubator to optimal temperature, say 30 to 35, or around 25 to 30 is also okay. And with continuous light exposure, that is very crucial here. Now, after a few days of incubation, you observe the petri plates for presence of colonies. Photosynthetic bacterial colonies, they typically exhibit a characteristic color that is green or red or purple, okay, depending on the pigment that is present. Now, you need a careful microscopic examination to be done here, okay. You don't want any other bacteria that is not a photosynthetic one to be growing, and you are carrying your all experiments with that. You don't want to pick up a contamination. So be thorough with your microscopic examination of the isolated colonies. Check for the cell size, shape, arrangement, okay, etc. Now, after that, you do colony morphology, try to get pictures. Then you can do gram staining, and after that, you can perform biochemical tests such as catalase, oxidase, etc. Depending on the type of bacteria you have isolated. Okay. Now, confirm it's confirm that they have the photosynthetic nature. Okay, that's the important one here. Now, to identify given bacterial species of a photosynthetic bacteria, you need to first do microscopy, colony morphology, and then biochemical test. For that, you need to refer Bergey's Manual of Determinative Bacteriology. In that, group 10 and group 11, you can see here an oxygenic photosynthetic bacteria. Various examples are covered under subgroup 1, 2, 3, 4, 5, 6, 7. Then, in case of group 11, that is oxygenic phototrophic bacteria, there are many examples covered. Okay, you can see the classification of cyanobacteria. Then there are I guess 5 to 6 subgroups for cyanobacteria as well. So depending on the biochemicals which are described in Bergey's Manual of Determinative Bacteriology, you can perform the identification test. Okay, now moving towards the viva questions. So the first is define photosynthetic bacteria. Very easy and basic questions. So photosynthetic bacteria, they are single cell prokaryotic organisms that can harness light energy to produce their own food similar to the plants. Okay. Now, unlike plants, they are anaerobic and they don't require oxygen for survival. Okay. Exception here is cyanobacteria. Okay. Others, they are anaerobic. Now, are there any photosynthetic fungi or yeast? This is silly question but yet tricky. Many students they just keep on thinking that are there any photosynthetic fungi? So no. When we are talking about photosynthetic bacteria or phototrophic bacteria, it means we are just dealing with the bacteria, not fungi or yeast. Fungi and yeast, they are eukaryotic, single cellular, unicellular and multicellular structures, and they are heterotrophs. Okay, that means they will always obtain their energy carbon from the organic compounds which are produced by other organisms, and they lack the key cellular structure and pigment that is needed for photosynthesis, that is chloroplast. Okay, so this is the reason there are no photosynthetic fungi or yeast. Now, why are purple non-sulfur bacteria generally preferred for isolating photosynthetic bacteria in lab? Because they have simpler growth requirements compared to the others, and their distinct coloration, that is the purple color, it simplifies the identification in the culture. Okay, so that's the reason why mainly purple non-sulfur bacteria they are preferred. Now, how can the experiment be modified to isolate oxygenic photosynthetic bacteria? Now, to isolate oxygenic photosynthetic bacteria, the isolation medium needs to be modified. That means you need to include a bicarbonate buffer system that allows them to utilize carbon dioxide as a carbon source. Then next question is, what potential application do photosynthetic bacteria hold in the field of biotechnology? So the potential applications are they are used or now they are being explored for bioremediation and biofuel production, okay, alternative energy source. So that's the reason why they are nowadays being explored more, more research is done. Then discuss some challenges that are faced in isolating and studying photosynthetic bacteria. You need to maintain anaerobic conditions, absence of oxygen if you are trying for anoxygenic photosynthetic bacteria. Okay, then differentiating between various photosynthetic bacteria, based solely on colony morphology is very difficult, because you just see them growing as a algae, okay, or green and purple color. So you can't just identify them based on their colony morphology. Okay. Now, can you explain the role of light in isolating photosynthetic bacteria? So the light, it acts as energy source for photosynthesis. That's the reason why we need illuminated growth chambers, which provide 24 hours light, okay, while incubating your culture. That's the reason why uh, it is easy to differentiate photosynthetic and non-photosynthetic bacteria in a given mixed culture. Okay. Now, how does the composition of BG11 medium supports cyanobacterial growth and the photosynthetic activity? So, actually, you need to study the media composition here, because it provides essential nutrients, minerals like nitrates, phosphate, trace elements. There are, I guess, 4 to 5 trace elements which are required for cyanobacterial growth, and you need to add those in your medium. So that supports the growth and the photosynthetic activity of cyanobacteria. Then next, what are some common pigments which are found in photosynthetic bacteria, and how do they enable these microorganisms to capture light? So photosynthetic bacteria, we have seen they contain several common pigments, including bacteriochlorophylls, carotenoids, phycobiliproteins, etc. Now, bacteriochlorophyll, they absorb light primarily in the infrared and certain visible range, allowing these bacteria to utilize wavelength of light that are less accessible to plants and algae. Whereas carotenoids, they absorb blue and green light, protect the photosynthetic apparatus from oxidative damage, while also contributing to the light capture. Whereas phycobiliproteins, which are found in cyanobacteria, they absorb light in orange to red spectrum, effectively broadening the range of usable light. And therefore, these pigments, they enable these all other types of photosynthetic bacteria to efficiently capture the light, convert the light energy into various environmental conditions, and that supports their metabolic process and growth. So this is about common pigments and how it enables the microorganisms to capture light for photosynthesis. So next is, how do photosynthetic bacteria adapt to various light intensities in their natural habitat, and how does this relates to their growth requirements in laboratory conditions? Now, the various light intensities that are present in environment and in lab conditions, they may differ. So the photosynthetic bacteria, they adapt to varying light intensities by modulating the synthesis and composition in their pigments. Okay, so these photosynthetic machineries, they optimize light capture, okay, that they can capture, and the energy conversion. So they can produce different types and amounts of bacteriochlorophylls, carotenoids based on the available light, and in low light they increase pigment concentration to capture more light, okay, while in high light or high, if the high level of light is present, then they adjust to prevent photo damage. Okay, so the bacteria, they are very clever, they have very good machinery to protect themselves and thrive in a given condition. So these adaptive strategies are replicated in laboratory conditions by adjusting the light intensity, ensuring the optimal growth and photosynthetic efficiency. Okay, that's the reason why you can easily isolate photosynthetic bacteria in labs also. Then can you discuss any specialized adaptation of photosynthetic bacteria that allow them to thrive in extreme environments such as hot springs or saline lakes? So yes, photosynthetic bacteria in extreme environments like hot springs or saline lake, they develop specialized adaptations such as thermostable enzymes. They use unique pigment composition. They have efficient light harvesting complexes. Now they possess robust cellular mechanisms to maintain functionality in high temperatures, high salinity. Okay, and these adaptations, they help these photosynthetic bacteria to thrive under harsh conditions. Okay, And the last one is what is difference between oxygenic and anoxygenic photosynthesis. So already this one is explained in my slides, so you can watch it and write down your answer. So be prepared for your practicals. Most important Viva questions or the preferred Viva questions I have discussed here. So I hope this video is helpful. Do like my videos, do share these videos with your friends, and do subscribe to my channel.